Welcome to the workshop for the ProgPal meeting for 2020. So I'm Graeme Lloyd, the name on the right here, but this is a joint effort between myself, Bethany, who you will no doubt meet at some point during the workshop, and Alex Dunhill as well. So I'm going to deliver this talk, but it was mostly written, it was written by really Bethany and myself, so we're the ones to blame if you have anything that you're confused by. So generally speaking, this is not ideally how we would deliver this workshop, but hopefully it will still work out reasonably well in the end in terms of giving you some general overview of sampling bias in the fossil record and some specific tools for how you might combat that with your own data. So just to give you a sort of overview of the structure of the workshop, so there's a separate talk that Alex composed for a different meeting previously that we're going to make available to you. So that gives you a nice overview in general about sampling in the fossil record. There's going to be this talk, which will probably have some overlap with that, but we'll also have some more in-depth stuff about methods. We also plan to make the slides for both of these talks available to you. Then on the GitHub, which I'm sure Bethany will provide you a link for, there's going to be some R scripts that you can work through for some hands-on with the methods, or at least some of the methods we're going to talk about. And then once you've done those, there will be the drop-in session on Friday the 12th of June. So the idea is that you attempt to watch the talks and do the scripts beforehand so that we're able to help you with things that you may not be sure about. You will not have enough time in the hour-and-a-half window to watch both of these talks and do the script, so it's, definitely try and do it in advance. Apparently there's also a Discord channel. I'm afraid I don't know much about that, but hopefully Bethany has informed you in advance, and you'll also be able to forward questions through the organisers to myself, Bethany, or Alex about what you will encounter on the workshop. So as I said, there'll be some overlap between this talk and the one Alex has, but broadly we've also organised it into three sections. So the first, which will be colour-coded, it would be sort of core concepts about sampling bias. Talk a bit about major sources of data that you could be using to do these kinds of things, and you can also contribute to some of those databases yourself too. And then we'll get more into the various different methods in the last third of the talk. So this slide I think appears in Alex's talk as well, but it's a nice sort of conceptualization of the fossil record in general. So ever since Darwin, of course, we've known or appreciated that the fossil record is incomplete, and therefore any kind of inferences we make from it, we have to be somewhat careful in how we do that. So we could conceptualize this sort of giant grey box as the the truth, the reality of the entire history of life. There's a smaller white box that represents the amount of that got fossilized, and then an even smaller box with the dashed line here, which is what might really be called the fossil record, because of that record word implies that in some sense it's been recorded, and more typically, I think formally, we'd think of that as a fossil that's been described and published somewhere in the literature. So we can then think, if you like, of the x-axis here representing effort. What we're really trying to do is push that black ball up the hill, increase our knowledge of at least the potential fossil record, and hope that is enough to get us towards understanding the history of life in terms of that grey box, even if the ultimate values are unknowable. So that effort is one way to consider how we can make the fossil record better, but sampling in general as a concept has many different facets to it. So effort's kind of represented on the left here, but you could also think of the number of scientists towards the top left there, the number of papers, the bottom left, the amount of money maybe in terms of grants if we want to be very ruthless in the top right, the amount of time spent on something bottom right, or just the number of fossils, which is what's shown on the right here. So these are ways to conceptualize sampling, but there are also ways to measure sampling. So as we'll see when we do the scripts, sometimes what you want to do is know whether or not sampling may be correlated with, and therefore perhaps driving whatever the sort of biological thing you're really measuring is. So conceptualizing it and measuring it is often a good start, although, as we will see, it's not always perfect in how we do these things. So it's important to state that regardless of how we do measure it, sampling will vary, and it can vary in lots of ways. So in particular, it tends to vary both spatially and temporally. So there are many examples we could use to show this, but here's one from a paper Bethany and Alex have coming out soon in Proceedings B. So here we're looking at the fossil record of tetrapods, going from the late Permian on the left to the middle Triassic on the right. But you'll appreciate, just looking at this quickly, that the coloured dots that represent our samples aren't continuously from the same spaces, or the total number of dots in each time window is very different in terms of its total count. So because of this, it creates a sort of complexity to what we're doing. We might not be able to sample a region we particularly want to, or a time bin that we particularly want to, and that can affect whatever it is we want to infer from that data. Sampling will also, of course, vary by clade. So here's an example from one of my papers. So some work I did with Matt Friedman a few years ago now. So this is what we might call a collector curve. So our x-axis here represents historical time, and our two curves represent the cumulative number of species that are named. So here we can see that Great British Fishes, which is what the GB stands for, have something like a convex curve. So we seem to have already named probably most of the ones that we're ever going to be able to in Great Britain. But by contrast, if we look here at the Mesozoic dinosaurs at the global level, we see this is a concave curve. It's sort of rocketing upwards, and I'm sure that's, even though it's a few years old, I'm sure we're still in that steep phase now. So this already tells you we've got many more dinosaurs to find, if not many more British fishes, and of course there's other ways we can consider sampling to vary across clades, and so any comparisons we make between clades, again, sampling something we might want to think about. Sampling also will of course vary anatomically. So the biggest way that's the case, of course, is that soft-bodied animals will generally not get preserved in the fossil record, or will require exceptional preservation, but of course everything has soft parts really of some form, and so oftentimes we're not sampling those for the species we do have fossils for. Instead, we largely have a fossil record of hard parts, so bones and shells, etc. But we can also break down preservation within a particular group by anatomy. So here is an example from the fossil record of birds, and we see here the long bones that are part of basically the wing or the leg are the most likely things to turn up in the fossil record for birds, whereas things like skulls in the bottom left here tend to be much less common, which is unfortunate if those are the parts of the anatomy that are most diagnostic or are most relevant to whatever it is you want to measure. So, for example, we could use birds here and say, well, the limbs would be useful for us to know about, say, locomotor diversity, but the skull would be useful to know about, say, dietary diversity, and this is something we always have to consider in terms of our own data. So, of course, the reason why this is a concern to us as paleontologists is that sampling may be an explanatory variable for things that we're actually interested in, and so what we might want to know about biology might not actually represent biology, but just our opportunity to sample. So this has obviously been appreciated for a long time in paleontology, but probably a sort of a key paper in terms of setting the direction of the field towards the present day was David Rout's paper in 1972 in Science. So in the latter half of the 20th century, paleontologists started putting together compendia of all the fossils we know and drawing out sort of these crude diversity curves, just counting often large level taxonomic or higher level taxonomic groups such as families and genera here, which are the three curves at the top of the plot. And these broadly show this sort of similar pattern of this relatively steep rise out of the Cambrian towards some kind of Paleozoic peak, perhaps in the Devonian, then something of a gentle drop-off towards the Permo-Triassic, and then after that a steeper rise towards the present in terms of diversity. So at the bottom there we have instead sedimentary rock volume. So that's representing the amount of rock available to sample fossils from in the Phanerozoic. And we can see that broadly that has a very similar shape to it to our diversity estimates. So this is problematic to us as paleontologists because it may mean that what we want to know about, which is how diversity changes over time, isn't really telling us that. Instead, we're just seeing the amount of available rock over time being reflected in our diversity estimate. But Rout's paper wasn't purely negative. It wasn't just pointing out this is a problem. It also offered two important solutions. So the first of these was modeling. So you could in some way regress out the signal of sedimentary rock volume. And the other was subsampling, which is basically setting an equal playing field for your the samples that you do have and looking at the diversity you get when you do that. So by far the more explored of these is the subsampling metric, so that's what we'll get to more when we get to the methods section. Modelling has been relatively unexplored, and unfortunately most of the methods generated so far have somewhat fatal, I would say, features to them. But I feel like maybe in the future someone will come up with a better modelling approach as well as a subsampling approach to deal with these things. So even a sitting president can realize that sampling is a critical kind of aspect to our patterns. More importantly, I guess, as paleontologists, what we want to know is when we have an absence of fossils, whether or not that's a real absence, or if it just reflects absence of preservation instead. So that's the thing we generally need to think about with our data, but it's not always easy to do so. So a critical feature, though, is that when we start to think about sampling bias, if we come up with methods to hopefully remove it, we can see that it can dramatically change our perceptions of the history of life. So on the left here we have one of Sepkoski's classic curves. So it has features, for example, of the Paleozoic plateau. So diversity seems to reach this level somewhere in the end of the Ordovician that it stays at until the end of the Permian. And then after the Permo-Triassic extinction we get this sort of steep rise towards the present. And on the right here is one of the attempts to do a similar thing. It's a different taxonomic level and it's much more data because it's two and a bit decades later, but we can see we get quite a different pattern looking at this data. We get sort of a peak in the Devonian instead of the, or the early peak is in the Devonian instead of the Ordovician. We see sort of somewhat depressed diversity, including a sort of late Carboniferous low, and we don't see such a steep rise at the end from the Permo-Triassic to the present. So this accounting for sampling can really change our perceptions of the fossil record and the history of life. So it's important, though, to realize what sampling bias really means and why we are specifically worried about it as paleontologists, and how it's different to other ways of dealing with sampling bias already kind of inherent in statistical analysis. So to show these points, what I've done here is taken a data set from the literature. So this is from Roger Benson and colleagues' paper looking at dinosaur body size, published in Paleontology just a couple of years ago. And I've basically created an alternate history of the order in which we encountered the fossils and then measured different aspects of body size along the way. So effectively, as we go from left to right here, we start out with one dinosaur, which is going to be our average dinosaur. It's also going to be our largest dinosaur, so our maximum value, and our minimum dinosaur, our smallest dinosaur. But as we add additional dinosaurs, those values are going to change, and they're going to separate, which is what we see as we go from left to right here. Until at the right-hand side of the plot we get to what we knew in 2018. So we have, I think the largest dinosaur at the top right there is Argentinosaurus, and then the smallest dinosaur at the bottom right is one of the Scansoriopterigidae. I can't remember which one off the top of my head. So the important point here is that we track the mean, shown in black here. We see that can go up and down as we increase our sample size. So if we wanted to do something like compare the average dinosaur with, say, the average crocodile, because it's the cystoclade basically, to dinosaurs. We wouldn't need to do anything special like what we're going to encounter in this workshop, because if we're doing something like a t-test, it already accounts for the fact that you have error around your estimate that's related to your sample size. So we wouldn't need to do that for the mean, but if we're looking at the maximum here or the minimum, we see there's a special feature that is essentially every time we pick up a new dinosaur, or we estimate the mass for a new dinosaur, it could be the largest dinosaur ever and things could increase for the maximum dinosaur, or it could be the smallest dinosaur ever and things will decrease for the minimum dinosaur. But the important point then is that the maximum and the minimum here have a monotonic relationship with sampling. So effectively it's asymmetric rather than symmetric, and so as we sample more dinosaurs we would expect the maximum to increase and the minimum to decrease, rather than have symmetric error, the kind of thing we expect when we're doing regular statistics because we're assuming a normal distribution, where we could underestimate or overestimate whatever the value is of interest. So it's this monotonic relationship that is the key thing to consider when we're thinking about sampling bias. Anything that has this monotonic relationship where the more you collect of a thing, it can only cause an increase or only cause a decrease. Those are the types of things we want to worry about in terms of sampling bias, and that's why we'd want to use the kind of specialist techniques we're going to encounter in this workshop. So here, by the way, is just the code if you really want to repeat what was there for the previous slide. I'm not going to talk through this, but if you want to have a go yourself, you can play with that data, and it's also available on the GitHub. So next we're going to talk about data. So it's obviously you could produce your own data sets for anything you're interested in, but if you want to do sort of many of the kind of larger analysis we tend to want to do now in paleontology, so major macroevolutionary analyses, so say at the global scale if you want to look at dinosaur evolution, realistically you can't build your own data set for that. You've got to use existing databases along the way. So briefly, I could talk through here are some of the major databases. There are definitely many more. So I could think of, like, for example, the Now Mammal Database or MyoMap. But we have, for example, at the top right here, the Neotoma Database. So this is, I think, largely focused at the moment on North America and plants and mammals. The bottom right we have the Geobiodiversity Database. This is a major Chinese effort, so it's intended to ultimately be a global database, and it's sort of section focused in its aim, so it's sort of more focused towards marine macroinvertebrates. We have the Neptune database on the right here, which is based around the major ocean drilling projects that have been in existence since, I think, the 1960s. So that's if you work on marine microfossils or nanofossils, that's an important database. iDigBio at the bottom left here is oriented around museum collections. Then we have the Paleobiology Database at the top left here, which is what we're going to focus on for most of this workshop. And then we have GBIF, which is assimilates a lot of different data sets together. So the important point, though, is that all of these data sets are they're often community-led, but they can be very inconsistent in terms of where the data come from and how well it's been entered, depending on the sort of historical aspects of those databases. And many of these are actually have quite long histories. So I know, for example, the Neptune database originally started out with punch cards, so that gives you an idea of how old it is. And so there's huge variety in the data there and huge inconsistency. So we need a lot of these databases to answer a lot of the questions we might have as paleontologists, but there are a lot of factors to consider in how they were constructed that are going to be important for what you want to do with them. So as I said, in this workshop we're going to focus on the Paleobiology Database, and that's because as organisers of this workshop that's largely what we've all been using. So it's the one we're most familiar with, but you shouldn't take that to mean it's definitively the best database. Certainly all of these have strengths and weaknesses. So it's important to stress, though, that regardless of which database you use, we would definitely also encourage you, if it's a community effort, to get involved in helping enter data, maintain data, etc. So I know Bethany and I would be happy to chat to you if you're interested in getting involved specifically with the Paleobiology Database, because that's the one we are both involved with in terms of entering data. But there's a couple of factors to consider here. So given they are community-led, if you want to use them for data, it's obviously nice if you also contribute to them, so you can feel like you're giving back to them. But probably if that's not motivation enough, it's also important to state that if you really want to understand that data, it definitely helps you to be involved in data entry. If you do that, you will realize exactly what a collection means isn't very well defined in the Paleobiology database, for example. And so that will mean you will do a better job in terms of how you treat that data when you are downloading it, analyzing it, etc. So definitely we're happy to chat to you about becoming members if you want to do so. It's just a form you fill in on the site. It's pretty easy to do, and then you just have to wait for a bit to get a password, and then there's various sort of helper videos to get you started as well. So as I said, we're going to focus in this workshop mainly on the Paleobiology database. So recently it acquired this nice front end that can help visualize everything in it. The navigator you'll see if you go to the home page. And this specifically is showing us all of the Phanerozoic records in the database. So you can see straight away there's a lot of data in there. We're approaching now 1.5 million fossil occurrences in the database. It covers everything taxonomically. You can have plants, for example, microfossils, dinosaurs, mammals, etc. And the colours of these dots represent geologic time as well. So you can see there's some good coverage, but also variable coverage, both in terms of time and space, and there's still some white you can see in there of gaps where either we don't have data entered yet, or there just hasn't been fossil collections from those places. So you can imagine why the interior of Greenland is white, for example. But there's also, I should say, we're up to now 420,000 taxa are entered in the database, so there's a lot in there, but it still definitely requires more in terms of getting to be more complete, and it depends on the group you're looking at as to how complete things are. So again, if you're involved in the entry of the database, you'll have some idea about that. If you've been downloading data and have something else to compare it to, you might also have some idea about that. But there are definitely areas where more work is needed, and if that's you, we can certainly sort of help direct you to making that database better. And of course, because it's a community effort, you're helping out paleontology in the broader sense, so you're pushing that black ball from the first slide further up the hill, basically. So there are other useful databases that we should consider in terms of the other aspect here, which is how we might measure sampling. So we're just going to look at one here, which is the Macrostrat database. So this is started by Shannon Peters. So he came up with this concept of macrostratigraphy, which is basically a way of dealing with rock units the same way we deal with taxa. So he treats them as gap-bound packages, so they have a sort of start in terms of deposition and an end in terms of the cessation of deposition that can be treated exactly the same way you treat the origination and extinction of a fossil. But this is also basically going back to that rock paper, getting us a handle on the amount of available rock that could be important for understanding biases in sampling. So here's sort of the map you'll get again on the front page for the Macrostrat database. So even though it's got pretty good coverage, there's the only sort of real white in there is the Arabian Peninsula. If you start to zoom in, you'll realize that the resolution varies dramatically globally, unfortunately. So we're not yet at a stage of having a truly good global geologic map, basically, that we can use in terms of available rock. But this does exist, and it may be good enough for whatever it is you're interested in. Certainly places like North America are very well covered, because that's where the whole database began from. Other places will be less so. But that's another important database to be aware of. So you may have already realized this. Of course, you will all have your own individual projects that you've been working on to for whatever it is you're interested in, you almost certainly will find, because it's from the fossil record, that it's in some way biased, and that's something you might worry about in terms of whatever your questions are about that data. So the important thing is not to worry too much about that. That's why this workshop exists. So there are lots of ways we can start to think about how we might deal with sampling bias, and get a way towards methods that might help you remove sampling bias, or actually even just address whether or not it really is a problem for the group that you're interested in. Okay, so we're going to shift gears now and actually look at what some of these methods are. But before we get into any details, it's important to state that almost everything that you're about to encounter wasn't developed by paleontologists or for paleontologists. Instead, a lot of it comes from the field of ecology. What ecologists do is very different to what we do as paleontologists. So this photo is meant to sort of emphasize that. So an ecologist might do something like this. They build these sort of quadrats. They would drop them somewhere in a meadow, and then they would sample everything inside of the say something like meter square that that quadrat represents. Now the reason why they do this is to make sure that their sample is unbiased. Ideally they would throw it without looking, for example, and it would stop them from only picking, say, the pretty flowers in this case, or only looking at the insects they like, or whatever it is. So as paleontologists, obviously immediately you'll realize we can't really do this ourselves, and that means there's probably bias straight away in our samples that is different to what ecologists have. But also, of course, if we dropped a quadrat on large parts of the rock record, there'd be no fossils in it at all, and so we have to sample things a bit differently. So it's important, though, to keep in mind that these methods are assuming that your data might look something like what an ecologist would get, but in practice what we have can be very different, and so that there can be bias in our sampling corrections as well as our data. So let's consider probably a relatively simple example in terms of what we might be interested in doing. So here we have, let's say we've got three very coarse time bins. So we've got the Triassic, the Jurassic, and the Cretaceous, and there's something we want to measure about them. So probably what we'll focus on for most of this talk will be something like taxonomic richness, so just the total number of genera or species, whatever it is we're interested in. But maybe we find the most species in the Jurassic, second most in the Cretaceous, third most in the Triassic. But if we look at our data, we would see we actually also have the most fossils in the Jurassic, second most in the Cretaceous, third most in the Triassic. So we could be worried then that sampling might be the reason why we see the richness counts we get, and so we might want to think about ways we can correct for this. So broadly then, there's two main ways we can consider of doing this. So the first might be extrapolation. So here we would try and take what we do know about our Triassic and Cretaceous samples and extrapolate it up to what we get from our Jurassic. Might have to let my cat out. Okay. So extrapolation is a sort of general concept in statistics, and generally if you remember your basic statistics, it's also one of the more dangerous things to do, unfortunately. So it's also important to state that you could extrapolate up to the level of your your largest sample, or you could try and extrapolate even further and make an estimate of what true diversity would be, but we can consider later on why that might not be a good idea. So extrapolation is one way to deal with this problem. The other way is to subsample your data instead down to the level of whatever your smallest sample is, for example. So tools to do this can include things like rarefaction and bootstrapping, but practically what you're doing is removing some of your data here from the Jurassic and the Cretaceous to try and create a level playing field. And another sort of name for this would be considering relative diversity, because we're not trying to get to absolute values of diversity. We just want to know which of various samples, in this case time bins, is more or less diverse. So you might ultimately be generating a time series, effectively. So I'm not going to talk too much about the mechanics of rarefaction and bootstrapping in this talk, because it's easier to show it, and that's done basically in some of the R scripts. So just go and check those out, I guess, for that aspect. So an important concept in general when we're thinking about sampling is the collector's curve or the species accumulation curve. So when we do these, we can have an x-axis that represents one of our measures of sampling. So it could be, for example, number of occurrences, number of fossils, whatever it is. And then the thing we want to measure on our y-axis. Again, here we're going to focus on richness, but going back to earlier in the talk, anything that might have a monotonic relationship with sampling could be something you could consider in exactly the same way, effectively. So let's imagine what we're doing here is we're going to a particular locality and collecting fossils, and we're ignoring basically anything that was collected there before. So our first field season we might go, we would find that generally every time we find a new fossil, there's a pretty good chance every time we pick up a fossil that it's a new species, effectively. So we would end up with this relatively steep curve in terms of as we add samples, add fossils, going left to right, we've got a good chance of finding a new fossil, new species rather, every time we do it. But as we go back perhaps in successive years, we start to find that we're just finding the same things over and over again. New species or new taxa tend to be relatively rare. And then by the time we've gone back several years, we find that we're somewhat asymptoting towards some kind of value that we might consider as being true diversity, but of course in reality it's not going to be true diversity, but the sort of potential in terms of preservation levels of diversity. So for example, you might want to consider this if we're thinking about gastropods. You've got some chance of capturing all the snails, but you're not going to capture any of the slugs, so we can't represent this as true diversity here. But generally, of course, we should be happy if we get to the level of asymptoting that we are getting a good handle on things in practice. Many things that we might want to do, we're more in that early phase and we're not sure effectively where our accumulation curve is going. So we can generate these curves logically. We would not normally record our data this way, so we wouldn't generate our exact discovery history, but we can regenerate them by resampling the data we do have, so randomly sampling from the fossils that we might have, and generating curves like this. And we do it 100 times or whatever it is, we'll get a curve that looks like this. And again, you'll learn how to do that in the DR script. So bootstrapping is one way of dealing with this, or generating those curves, and then you can pick some kind of equal sampling level to compare across your samples from. And this as a concept goes back at least to Foote in 1993, so this paper in Paleobiology where he was looking at trilobites, and he was looking at both species richness, so the number of taxa, and measures of morphological diversity, often termed disparity. So here is a useful plot that you would want to make anyway to check whether or not sampling might be a problem for your data. So what Foote's done here is he's rescaled his data 0 to 1. So on the x-axis we've got relative species richness. So the least species rich sample gets scored 0, the most species rich is 1, and everything else is stretched out in between. And then he's done the same thing for the relative sample size on the y-axis. So the smallest sample is 0, the largest is 1. And we see that even though things fall out on a sort of positively sloping line here, they don't fall perfectly on that line. There's clearly some variation in there. So even though there's a positive relationship between sample size and species richness, there's also some other signal in there that you might want to get at that is hopefully at least more likely reflecting something biological and the thing that we're interested in. So bootstrapping is a way of doing that. Again, the practicalities of that are best shown in the script, so I'll refer you to those in terms of how you might do it. And here he's sort of shown the results you get looking at disparity this time instead of richness for the trilobites. And on the left here is comparing the relative components from the Facopid trilobites and the Librostoma trilobites. So the same data is effectively plotted on the right here, but it's oftentimes a bit easier if you're looking at different components of a curve to plot them separately, because it can be hard to read across those. And we can see also another advantage of bootstrapping is that we can see we've got error bars there around our data, and bootstrapping is a good way of looking at what uncertainty you have in terms of your data. So one way to think about what bootstrapping is doing is it's creating a bunch of alternative sampling histories that you may reasonably have encountered with your data, and that those the variance you see in those is kind of what generates the error bars here, but also you can resample to the same level to allow basically sampling to be equal, and therefore in theory making a fair comparison between subsets of your data. So when we generate these sampling curves, and we can do this through either rarefaction or bootstrapping, we can end up with situations like this. So this is something I call the Ghostbusters problem, which is a reference to an old film. If you know Ghostbusters, one of the plot points of the movie is that you shouldn't cross the streams of the proton packs. But what I mean in this context is we can be in trouble if our rarefaction curves cross each other in the space. So all our sampling curves have to start at the origin. Basically, when you have 0 fossils, you have 0 richness or whatever else you want to measure. But they normally will diverge after that, but sometimes they can end up crossing later on in the space, as we see in this example here. So this creates a problem because what we want to do is have a fair comparison between A and B. So if we look at the top right of A and B, so where the curves terminate, we see that A has higher richness than B, but also on the x-axis it's gone further along, so it's a larger sample size than sample B. So we could pick some point indicated by the vertical dashed lines here where we can compare the two on an equal footing. But if we see at the left-hand one of those, we would infer at that point that B is actually more diverse than A. Whereas we look at the middle dash line, we'd say, well, actually, they're equally diverse. And if we look at the right-hand dash line, we would say, well, A is in reality more diverse than B. So if you want to know which is true, probably in this case, we would be happier that A is more diverse than B, and that's because we're able to sample far enough to see that they are clearly diverging. They wouldn't cross again in the space if we continue to sample, if we continue to increase things going to the right. But it's important to bear in mind that in reality we may not have got to that second dash line. We may have only sampled enough of our curve to be in the A is less than B part of this plot. So in general, what you want to do is make sure you generate plots like this, and again you'll do one in the script, and consider whether or not things are truly diverging, and therefore whether or not you can accept which is more diverse, or if they're a bit too close to each other, you might want to be a bit more hesitant about how you might interpret that data. If, for example, you never generated these curves, you just picked one of those dash line values and then made inferences from it, you could basically be in danger in terms of understanding the data. So another important aspect to this, and the reason why you might get curves that look such different, have such different trajectories, is another important concept, which is known as rank order abundance. So what we'll do here is each of these sort of bars represents one of the species in your sample, and the height of the bar represents abundance in some sense. So again, in reality, if you're an ecologist, it'd be the number of, say, daisies you count in your quadrat would be your abundance, but in practice in the fossil record, oftentimes we haven't physically gone to a location and counted every fossil we find. Instead, we're doing things like using a major database, and therefore the abundance might really represent the number of occurrences. So that's the number of different localities your species is known from. So once you have something that is considered to be abundance, you can then basically sort your species from left to right, where you have your most abundant species on the left, your most common species, and then you have your least common on the right, and of course you can have ties like we see in the middle here. So the shape of this rank order abundance is what decides the evenness of your sample, and that would decide things like the shape of a rarefaction curve along the way. So some important things to think about here. So first of all, this is not a very realistic looking one. Generally there will be a most common species that's much more abundant than everything else, and oftentimes you'll have a a long tail on the right of least common species. So for example, you might have lots of singletons where you've only got one fossil or one occurrence for many of your taxa. So In an ideal world, if your least common species has been sampled more than once, then you would know that you'd actually already hit true diversity, or at least the best that we can get in terms of preservation. Again, you might have all of your snails, but you would not sample any of your slugs. But generally, you'll find that you have your least common species will only be known once, and that implies there's more sampling to be done, or more sampling in terms of more species out there that are not sampled. So we can use this rank order abundance in several ways. So one way is to treat it like a curve, so mathematically as a curve, and then you can use calculus to basically ask for different areas under the curve, and that's another way to subsample your data. So this is the general idea of how what are called coverage-based methods work. So we could how the proportion of that area could be, for example, 40%, that you would call a quotient of 0.4. And you'd go from left to right, and then you would read off from the bottom a subsample diversity estimate. So importantly, this is going to be a relative estimate, and it will also oftentimes not be an integer, which can be a bit non-intuitive for people. So it will estimate, in this case, you probably get a diversity of, say, 2.53 estimated in terms of richness. And of course, you can vary that quotient along the way. So that's kind of a simplification of how coverage-based methods work. So there are many existing coverage-based methods out there. So this plot shows a couple of those. So we've got SQS on the left here, specifically the top left. We've got TRIPS on the right here, but there's also things like char1 and squares, which is something we'll encounter in the scripts. So some of these coverage-based methods can be preferable to things like rarefaction, and so the plot on the left can show why that might be the case. So the bottom left here is regular rarefaction, and as we see, the sample size, which is what each of those lines represents, the number at the end of it, as that gets smaller, we see that we end up with quite a flat curve. So that's shouldn't be interpreted literally as that there's no diversity change. It's really more that we don't have a big enough sample size to see good variance in the data. Whereas if we look at what SQS is doing at the top left here, we see regardless of our quotient size, which is, again, you can think of that as the area under the curve we're asking for, we see very, very similar relative diversity shapes to those curves. So that's kind of a nicer feature in that we're going to see more variance even if our sample size is not very large. So TRIPS on the right here has had something of a mixed reception, but it's an extrapolator as well as a coverage method, so it's trying to estimate true richness, which can obviously be a tricky thing to do. And the example from the original paper, which is again part of the reading list, is looking at Mesozoic dinosaurs. So not going to talk too much more about that, but that's one of the papers you can read about. So these methods can be, again, really important in terms of how they can change our perceptions of what's going on with diversity once we consider sampling bias. So here's an example where things get changed completely. So on the left here we have raw tetrapod diversity, that's what the black line is in here, and we're looking at an interval through the Carboniferous to early Permian in tetrapods. It's a paper by Emma Dunn and colleagues in Proceedings B. So the red boxes here are the same time window, and then the blue box shows on the left here a collapse associated with a rainforest collapse going from this Carboniferous to Permian transition that in the raw data seems to also show a collapse in tetrapod diversity. But when we look at the coverage methods, which is what you see on the right here, instead the same time window is actually a peak in the diversity in the time series. So once we account for sampling bias, at least in this attempt, it flips basically our perception of what's going on there. So instead of tetrapods tetrapod diversity being strongly affected by this loss of Carboniferous rainforests, we actually see a peak in diversity when we look at the SQS method. So obviously there are many of these things out there, and so a good question would be, well, which is the best of those? So there's a couple of papers, again these are in the reading list, that have attempted to look at this with things like simulations, but also some empirical examples. So the figure here is from the close paper, but there's also a much more recent paper by Alroy in Paleobiology. So each column of this figure is a different shape to that rank order abundance curve. So on the left here is flat, which is pretty unlikely, so that's suggesting abundance is the same for all of your species. And then the next three columns are all log normal distributions, so you'd have a lot of your common species, and then this long tail, increasingly long tail of rarer species here. So we see in each case we've got the red line representing face value count, so that's if we didn't do anything to kind of correct for sampling bias. And then we've got three, I think those are all coverage-based methods in the different colours. So we can interpret a few things from this. So first of all, as we go left to right in each of these plots, we're increasing our sample size. The dashed line at the top is true diversity. So obviously, ideally, we want to hit that very quickly. And we see we do that on the left when our rank order abundance is flat, with most of our methods get there very quickly relative to face value counts. But of course, that's not very realistic, and so the more realistic distributions, the three right-hand values, we see they don't get there quite as quickly, although they do tend to do better than the face value counts. So this doesn't perfectly tell us which is the best ones. I would recommend reading the Alroy paper as well, so he comes up with some new clever tests to think about these things. And there are obviously also other methods that aren't considered in the close paper that Alroy does consider. So those are things to appreciate as well. But generally, this is going to affirm the fact that when our sample size is small, our ability to say things is going to be more uncertain. So there are other methods that unfortunately we can't cover in detail in this workshop, but it's important for you to know about, and this is another one that you can read up about through the reading list. So capture-mark-recapture is again it's based on an ecological technique. So in practice, this would normally be used to estimate population sizes in the wild. So let's say you're interested in the population of a particular species of bird. You could go out, collect birds. When you collect one, you can put some kind of mark on it, say a tag. Then you release it, you go and collect another bird, put a tag on it if it doesn't have one, but eventually you're going to start to recapture the same birds, and you'll see that the tag is already in place. So effectively, the population size is very small. You would find that you're recapturing things you'd already marked very frequently, whereas the population's large. You would finding birds without marks already on them more commonly. So that may seem not very intuitive to how do we apply that to the fossil record, and so I suggest to sort of find out about that, you go and read papers by Nichols and Li Xiang Liao, as they explain it better than I can in the time we have here. But importantly, these can be really nice in that they can help us estimate not just estimates of true diversity, or at least true diversity in terms of preservable diversity, but they also get us towards things like extinction and origination, which is what you see in the top two rows here, but also measures of how sampling can vary over time, which is what you see on the bottom row here. So they're really nice, but the disadvantage to them is that generally they're only going to work when you have a high resolution temporal data set with pretty abundant groups. So effectively they're great for things like marine invertebrates, like the bivalves and brachiopods we see in this figure, but they're not going to work at all if you have much coarser, much less well-sampled data sets like a lot of vertebrate groups are. So another field that was the one that I said was underdeveloped, suggested by Raup, is to think about modeling, so regressing out that data and perhaps getting some kind of residual diversity estimate. These are a couple of papers that have explored that and some of the reasons why we might not be so confident in the results we might get from those. So on the left here is a paper by Brocklehurst from 2015, so I think it's all simulated data. So he considers a bunch of different metrics. So the black in here is true diversity estimate. Then he has the taxic diversity estimate you get after simulating sampling, so that's the lower down grey line. But then he has two ways you might correct for that. So one is phylogenetic diversity estimate, the PDE, the red line in here, so that's trying to fill in the gaps using a phylogeny. And then the blue is the residual diversity estimate. So you can go and read that paper for more details. And then the one on the right is a paper that includes both simulated and empirical data sets. And again, it looks at using these sampling proxies, so things like the amount of sedimentary rock. I think more specifically, it looks at the number of formations, which is another common proxy that's been used. And they suggest that basically if we do that, you can often throw out some of the original data too. So it can be a bit dangerous to do in that you can lose some real biodiversity signal by trying to regress out sampling that you don't really want to do. So I think there's lots of other issues with the sampling proxy correction methods as well as these, but we're not going to teach you these in the workshop, but generally we probably wouldn't recommend them anyway. I suspect there may still be a better way to use modeling, but that's yet to exist basically. So it's important to also consider phylogenetic trees. So that was briefly considered in the Brocklehurst paper from the previous slide. But if you do work on vertebrate groups, for example, you're likely to have a good phylogenetic control of how your species are related. And more generally, of course, if most of the things we're applying, we're assuming our species are independent units, but of course they're not. They're connected through a phylogeny. So a more recent kind of development in this field that's been incredibly important is what's called the fossilized birth death model. So that was came up with by Tanja Stadler, effectively a mathematical biologist. But it turns out to be incredibly useful and important to us as paleontologists. And we can use it to do lots of really important things. So it can help us, again, think about things like origination and extinction in the context of a phylogeny. But phylogenies are also really important in terms of showing us where we might expect to sample lineages that we might not actually have been able to sample. And so it can help us get to sampling time series as well. So it could be useful to combine this information effectively, not just your occurrences distributed through time, but any kind of phylogenetic information you might have about those as well. So technically, I think this can be used to look at things like richness, although it would be more in the context of a number of lineages with a phylogeny. But I think that's relatively unexplored at the moment. So that could be something interesting to look at in future. And finally, probably sort of the most recent stuff and the most probably making some really important points is thinking about the spatial aspects of sampling. So almost everything we've looked at so far assumes you have a time series you're interested in, so looking at diversity changing over time. But as we mentioned, sampling will also vary over space, and that can affect whatever it is you're interested in. So a series of papers by Roger Close, including this one on the left here from Nature Communications, have looked at this spatial aspect to sampling. And so these two figures show a way we can consider how spatial sampling might affect things using what's called a minimum spanning tree. So you have a series of locations in space, but it's important to consider if you're sampling quite far apart, you'd expect things to be much more different, and so you would increase your diversity. Let's say, for example, if we're sampling mammals, if we just sampled North America and Europe, we'd probably find our mammal faunas would be pretty similar, whereas if we sampled Africa and Australia, we'd find they'd be very different, and we'd expect more diversity if those samples come from further apart. So the minimum spanning tree here is an attempt to account for that, but I think more broadly, Close et al. Would say that global diversity estimates aren't something we can really aspire to. Instead, we have to think about regional diversity because the spatial component is too important effectively to ignore in our data, and we can't sample uniformly in space and time, unfortunately. And then on the right here, again, is even more sophisticated techniques where we can use things we might know about the ecology of our species to predict where we should find them versus where we do find them. And so we can then compare sort of predictions under different models with what we actually observe empirically. So again, I'm not going to go too much into the details on these, but these are things you can read up about. And again, we don't cover them also in the scripts, unfortunately, but because they're a bit more advanced than what we're able to do. But these are all things you can be thinking about potentially in terms of your own data. So that's going to be the end of this talk.
But please make sure you, if you work your way through the scripts and the other talk, if you have any questions, please come along to the Zoom meeting and ask. You'll be able to ask myself and Bethany and Alex, I think, at that meeting. So yeah, that's the end of the talk.